One, and today we will be presenting on the immune system. Our group comprises of myself, Cheryl Alfred, Natalia Barnwani, Abigail Brewster, and Chevelle Cornwall. We'll begin today's presentation by having an overview of the human immune system. The human immune system is the body's defense system. It is made up of different organs, cells, and proteins working together to achieve one goal. The human immune system comprises of two main parts, the innate immune system and the adaptive immune system. The innate immune system is the immunity that an individual was born with, whereas the adaptive immune system develops as an individual's body is exposed to microorganisms. The innate immune system responds first when an invading pathogen is detected. It is made up of the skin, mucous membranes, linings such as the respiratory and the digestive tract, as well as the corneas of the eyes. These create physical barriers that protect the body against pathogens. This immune system becomes activated when an individual is born. The adaptive immune system is responsible for making antibodies that are designed to fight specific pathogens while protecting the body against them. These antibodies are developed by cells called B lymphocytes after the body has been exposed to a pathogen. Key components of the immune system. The immune system consists of both cellular and molecular components. The molecular components include antibodies, antigens, which are specific agents, and cytokines, non-specific agents. These components work together to coordinate an immune response. All cells of the immune system are derived from pluripotent stem cells in the bone marrow. Cellular components. Lymphocytes. Lymphocytes deriving from pluripotent stem cells in the bone marrow. They are then differentiated by passage through the thymus, which would give T lymphocytes, or will mature in the bone marrow, B lymphocytes. They possess the ability to recognize specific antigens, causing a specific response of the immune system. Lymphocytes account for 20% of circulating leukocytes. B lymphocytes are responsible for maintaining memory for antigen exposure, as well as secretion and elaboration of specific antibodies. Naive mature B cells are able to express only non-specific IgM and IgD, but after activation are programmed to produce specific immunoglobulins, IgA, IgE, and IgG and IgE. Mononuclear cells. The mononuclear phagocyte system has two specific functions: phagocytosis of foreign material and processing and presentation of antigens to T cells by antigen-presenting cells. Phagocytosis is important in non-specific clearance of foreign antigens. Antigen presentation is critical to the specific functions of the immune system in surveillance, memory, amplification, and antibody production. Antigen-presenting cells, or APCs, are specialized macrophages that are able to recognize foreign substances as antigens and process those antigens by enzymatic degradation. They are found in the skin, lymph nodes, and spleen. Granulocytes. There are 4 types of granulocytes, and they are neutrophils, eosinophils, basophils, and mast cells. Neutrophils are the most commonly occurring and frequent class of granulocytes. They are multilobulated cells that are drawn to sites of inflammation by cytokines. They engulf pathogens, destroying them with proteolytic enzymes. These enzymes are contained in cytoplasmic granules that are released on contact with bacteria or other pathogens. Eosinophils are bilobe cells that account for 2 to 5% of circulating granulocytes in the non-allergic individuals. Their proportion is usually elevated in allergic reactions and is briefly present in circulation. Mast cells and basophils are virtually indistinguishable microscopically and functionally, and differ in that mast cells remain positioned in tissues while basophils are present in circulation. Both mast cells and basophils are involved in the allergic response and bind IgE molecules to their surfaces. Molecular components. Immunoglobulins. They are glycoproteins that are produced by B cells in response to antigenic stimulation. They are the primary immune effectors and are responsible for the specificity of immune response. There are 5 classes of immunoglobulins: IgG, IgM, IgA, IgD, and IgE. Cytokines. Cytokines are small regulatory polypeptides that are responsible for communication among cells of the immune system. They are potent messengers that are produced by many types of nucleated cells, including lymphocytes, macrophages, epithelial cells, endothelial cells, and fibroblasts. Complement. Complement refers to a series of protein molecules that function in a coordinated cascade in response to antigenic stimulation. It is involved in both specific and non-specific immunity. Its role is to augment immune function. Complement is activated through antigen stimulation and under the direction of immune cells. And I'll pass you on to my colleague Chauvel, who would continue the presentation.
I'm Chauvel Cornwall, and I'll be doing the immune responses. There are 2 types, which are primary and secondary. Primary responses refers to any immune responses of the immune system that includes the production of antibodies or cell-mediated immunity. It occurs following the first exposure to a foreign antigen. At the beginning of the primary immune response, there are no previously produced antibodies. Hence, B cells are activated and differentiated into antibody-producing plasma B cells. Some of the plasma B cells develop into memory cells. The lag phase is a time period in which antibodies are produced against a particular agent of the primary response is long. Sometimes the lag phase can be as long as weeks or months. The secondary response refers to any immune response of the immune system that occurs in response to subsequent exposure to a particular agent. The cell's exposure may occur even after a year. Due to the presence of the memory cells, the lag phase of secondary response is shorter than the primary immune responses. IgG is a main type of antibodies produced, and small amount of IgM and IgA and IgE are also produced. A high antibody titer is produced during the secondary responses.
Hi guys, my name is Abigail Vista and I will be doing the mechanism of action along with the example of diseases and immune system dysfunction. So the human immune system is the body's defense system. It is made up of different organs, cells, and proteins working together to achieve one goal. The human immune system contains 2 main parts: the innate immune system and the adaptive immune system. The innate immune system is the immunity that an individual was born with, whereas the adaptive immune system develops as an individual's body is exposed to microorganisms. The innate immune system responds first when an invading pathogen is detected and as shown in the picture, it is made up of the cornea of the eyes, the mucous membrane lining of the respiratory and digestive tract, as well as the skin. So these create a physical barrier that protects the body against pathogen. It is important to note that this immunity is only active when an individual is born. So now let's look at the adaptive immune system. So the adaptive immune system is responsible for making antibodies that are designed to fight specific pathogens while protecting the body against them. These antibodies are developed by cells called B lymphocytes after the body has been exposed to a pathogen. So as shown as in the picture, we can see other cells along with the B cells, the T cells. So we have the T lymphocytes. So the T lymphocytes have memory cells that remember the pathogen, the T helper cells that activate other cells to destroy the pathogens, the cytotoxin cells that destroy the pathogens directly, and the regulatory cells that suppress the immune response in order to prevent overactivity. So now let's have a look at the examples of diseases and immune system dysfunction. The immune system dysfunction can lead to various diseases, each affecting the body's ability to defend against infections, such as an overactive immune system, autoimmune disease, immune system disorder, and cancer of the immune system. So let's do a breakdown. An overactive immune system, the immune system reacts to different substances in the environment that aren't normally harmful. For example, we have asthma. The immune system overreacts to triggers such as dust or pollen, causing inflammation in the airways and difficulty breathing. Eczema, a chronic inflammatory condition where the immune system reacts to allergens or irritants on the skin, causing itchy, red, and inflammatory patches. Allergic rhinitis. This is an inflammatory of the nose caused by an allergic reaction from indoor allergens like dust and pets, or sometimes other outdoor allergens like pollens or molds. This may cause you to sneeze, a runny nose, sniffing, and may cause swelling of the nasal passage. 2. An autoimmune disease. So, autoimmune disease occurs when the immune system creates antibodies that attack its own healthy tissues. So, type 1 diabetes. The immune system destroys insulin producing cells in the pancreas, disrupting blood sugar regulation. Rheumatoid arthritis. The immune system cells attack the joints, causing swelling, deformities, and inflammation in the joints. Lupus. The immune system attacks the body tissues, including the lungs, kidneys, and skin. 3. Immune system disorder. So we have the immune system. This is a dysfunction of the immune system. For example, we have the severe combined immunodeficiency, often called the bubbly boy disease. This is a genetic condition that prevents the immune system from producing essential immune cells, leaving the body highly vulnerable to infections. Then we have the DiGeorge syndrome, a genetic disorder where the thymus gland does not develop properly, leading to a shortage of T cells and a weakened immune response. So now let's have a look at cancer of the immune system. Cancer of the immune system, this is a type of cancer that starts in the immune cells of the lymphatic system. For example, we have the lymphatic lymphoma, a type of cell that starts in the immune cells called lymphocytes, causing them to grow uncontrollably. And last but not least, we have leukemia, a cancer that begins in blood formation cells in the bone marrow, leading to the production of abnormal white blood cells that interfere with the normal immune function. I will now hand you over to my colleague Natalie, who will be doing the recent advancement in immunology or the immune therapy.
Now I will be speaking about recent advancements in immunology or immune therapy. In recent years, the knowledge of immunology has expeditiously improved, and there are more advances to come in the near future. These advancements have had many factors involved, one which includes thousands of different monoclonal activities being developed that allows a huge variety of cell subpopulations being identified. Immune cells and their functional analysis are also a recent development that has had advancement. Some examples of recent technologies are single-cell analysis, image techniques, omics such as DNA, RNA sequencing, proteomics, and metabolomics data, and also newly designed animal models. All of these advancements can be put to use in many immune-mediated pathologies. However, in recent years, there have been successful achievements with types of immunotherapies, and it has been revealing new ways that are beneficial to humans for exploring and manipulating the immune system. Next, I will touch a bit on immunotherapy. In terms of recent advancements in immunotherapy, there have been some advancements in the field of oncology. There has been a large growth in immunotherapeutic strategies to treat cancer, which include immune checkpoint inhibition, chimeric antigen receptor T cell therapy, and cancer vaccination. Now, this is the end of Group 1's project. We hope that you enjoyed this presentation, and if you have any questions, feel free to ask. Thank you.